Ever get that feeling that there's this whole hidden world operating inside us? A super intricate biological machine we only get glimpses of.
Oh, definitely.
You're not alone. Today, we're pulling back the curtain, taking a fascinating journey into that inner world. We're diving deep into the realm of molecular biology, exploring some really compelling articles that highlight its most exciting developments.
Yeah, some really groundbreaking stuff.
Think of this as your express route to grasping the fundamental principles and the truly mind-blowing progress happening in this field right now, all without needing a PhD.
Exactly. We've looked at sources that trace the evolution of molecular biology, from its initial conceptual sparks right through to the cutting-edge technologies that are fundamentally changing how we understand life itself.
Okay.
Our goal today really is to extract the most crucial insights from this journey, those pivotal aha moments, and the powerful tools that have emerged.
And figure out what it all means for the future.
Precisely. Understand what they signify for the future of science and, well, health.
Okay, so let's start at the very beginning, or at least what feels like the beginning of modern biological understanding. Our sources point to some truly foundational work by a guy named Gregor Mendel way back in 1865.
Ah, Mendel, yes.
It sounds like he brought a whole new level of precision to the study of life.
He did indeed. Mendel's work on how traits are inherited was, well, revolutionary. What he introduced to biology was a quantitative approach.
Quantitative, so like numbers and measurement.
Exactly, a way of measuring and analyzing biological phenomena with a rigor that had previously been more common in fields like, say, physics and chemistry. Through his careful experiments with pea plants, he established the fundamental laws that govern how characteristics pass from one generation to the next.
And it's wild to think that this groundbreaking work was initially just myth.
Completely overlooked.
It wasn't until the early 1900s that it was rediscovered by several scientists independently.
Correns, de Vries, Tremacia.
That must have been an incredible turning point for biologists who, as our sources suggest, were looking for more than just describing and categorizing living things.
Oh, a huge turning point. Imagine the relief.
It must have felt like they finally had a real framework.
Absolutely. While Darwin's theory of evolution had already hinted at functional relationships in the living world, Mendel's work provided the concrete, quantifiable framework scientists were really craving.
Ah, okay.
This rediscovery wasn't just a historical footnote. It fundamentally shifted biology from being mostly descriptive to being truly experimental. It let researchers ask why and how with actual data, moving beyond just looking and naming.
It's so human, that desire to understand cause and effect. We always want to know why things happen.
Innate curiosity, right?
But when you look at something as complex as a living organism, figuring out those connections can feel like trying to untangle a giant knot.
That's a great analogy. Living systems are incredibly intricate, and cause and effect isn't always a straight line. It's messy.
So how do scientists deal with that?
Well, this is where the scientific strategy of focusing on simpler systems becomes so valuable. You break down those complex networks into manageable, reproducible components, shorter chains of influence.
Makes sense.
Think of those fundamental rules of nature that our sources highlight, like the genetic code itself, or the process of protein synthesis at ribosomes, or the operon system for gene regulation in bacteria.
Okay.
These are core relationships, sort of foundational rules that help us piece together the bigger, more complex picture.
And this shift towards a more quantitative molecular understanding, it wasn't universally embraced right away, was it? I recall our sources mentioning some back and forth about different approaches.
You're right. Not everyone jumped on board immediately. As molecular biology provided these increasingly detailed explanations at the molecular level, it naturally sparked discussions.
Like what?
Well, debates about whether understanding the individual components, the molecules, the genes, was actually enough to understand the whole system, the whole organism.
The reductionist versus holistic thing.
Exactly that. This ongoing dialogue between what we might call reductionist and more holistic viewpoints hasn't just influenced scientific research, but it's also shaped how the public perceives biology's applications, in medicine, agriculture, all that.
Okay, here's where it gets really interesting, because these molecular biology methods aren't just abstract ideas stuck in labs.
No, far from it.
They're actually being used to tackle really pressing real-world problems, from developing new ways to prevent and treat diseases,
Crucial work.
to engineering proteins with novel functions and even modifying the traits of plants and animals for specific needs.
Precisely. And as these methods become more common, more integrated into our, well, advanced society, as our sources point out,
People start asking questions.
it's understandable, right? Questions and even concerns arise about how they're being used. It's an incredibly powerful set of tools.
And with great power
Comes great responsibility, exactly. Like any powerful tool, there are ethical and societal considerations that need careful thought and, importantly, public discussion.
It's still kind of mind-blowing to think about how deeply this understanding of molecular biology, particularly how nucleic acids control the fundamental characteristics of a cell, has woven its way into our everyday lives.
It really has, often in ways we don't even realize.
Now, our sources make an important distinction between basic and applied research in this field. Can you break that down for us and explain how molecular biology fits into both?
Certainly. So, basic research is fundamentally driven by a quest for deep understanding.
Just know it for knowing's sake.
In a way, yes. The desire to know the underlying mechanisms of how life operates. Applied research, on the other hand, takes the discoveries made through basic research and uses them to create something of practical value.
Like a new drug or a better crop.
Exactly. A new medication, an improved industrial process, a more resilient crop, you name it. Molecular biology is absolutely foundational for basic research. It gives us the techniques to investigate life's core processes at the molecular level. But it's the application of these techniques that often captures public attention, leading to those tangible advancements in medicine and industry that people notice.
It's like the analogy our sources use with fire. We use fire all the time, right?
Yeah.
But most of us don't need to understand the complex chemistry of combustion to boil water or stay warm.
That's a perfect analogy. We're constantly benefiting from the applications of molecular biology in countless ways, even if we don't grasp the intricate science behind them every time.
Now, speaking of those intricate sciences, recombinant DNA technology gets mentioned a lot. What exactly is that, and why is it so significant?
Recombinant DNA. It's a cornerstone of modern molecular biology. It essentially allows scientists to manipulate DNA outside of a living organism.
Manipulate how?
Think of it like being able to cut and paste DNA sequences. Specific proteins, called restriction enzymes, act like molecular scissors.
That's interesting.
They cut DNA at very precise points. Then other enzymes, ligases, can join different DNA fragments together, creating new combinations, recombinant DNA.
Okay.
This ability to rearrange and analyze DNA has been absolutely critical for understanding how genes are organized and, importantly, what they actually do.
And that, in turn, paved the way for cloning methods, which I think most people associate with making identical copies of organisms like Dolly the sheep.
Right, that's the common association.
But it sounds like in molecular biology, cloning serves a slightly different primary purpose.
That's right. While cloning whole organisms is one application, often a controversial one, in molecular biology research, cloning methods are primarily used to produce large quantities of a specific pure DNA sequence.
Why is that useful?
It's incredibly valuable for studying how genes are regulated within cells. It allows researchers to really delve into the mechanisms of gene control, isolating one piece of the puzzle. This capability revolutionized the field.
Wow.
And as our sources note, it continues to fuel scientific discussions and, refinements of our understanding of how genes actually work.
It's fascinating that the discussion surrounding the impact of recombinant DNA
Right.
extend beyond just the scientific lab.
Mm.
Our sources touch on safety considerations, ethical implications,
Oh, yes.
and even economic impacts. It really highlights how these scientific advancements don't happen in a vacuum.
Precisely. These technologies have far-reaching societal consequences. It's natural, maybe even necessary, for diverse viewpoints, and yes, sometimes misinformation to exist outside the scientific community.
Yeah.
It's crucial to remember these are no longer just tools for academic scientists in ivory towers. They are being applied in a wide array of fields to address practical, everyday problems.
Okay, so we've covered some foundational principles in these powerful molecular tools. Now let's zoom in on something that feels very personal. The ability to unlock individual genetic signatures.
The unique genetic code.
It sounds like molecular biology has given us ways to really pinpoint what makes each of us genetically distinct.
Yes, and the key molecular biology techniques that allow us to do this are things like restriction fragment length polymorphism analysis, RFLP for short,
RFLP, okay.
polymerase chain reaction, or PCR, which most people have heard of now,
Definitely.
and DNA fingerprinting. These methods all rely on a fundamental fact. Each individual, except for identical twins, inherits a unique combination of DNA,
The code that makes us, us.
the very code that dictates many of our physical characteristics, yeah.
So by isolating someone's DNA, we can essentially read parts of that code and potentially identify or even predict certain traits within a group.
Exactly. Let's take a closer look. RFOP analysis uses those restriction enzymes we talked about, the molecular scissors.
Right.
Because individuals have slight variations in their DNA sequence, these enzymes will cut the DNA in slightly different places, producing DNA fragments of different lengths.
Oh, okay.
When these fragments are separated based on their size, maybe using gel electrophoresis, they create a unique pattern of bands, a sort of fingerprint. And this pattern can be used to identify genetic variations or even mutations linked to particular traits or diseases. Think of it like cutting a long string at slightly different places for different people. The resulting pieces will have different lengths.
Makes sense. And PCR, polymerase chain reaction, that's the technique we've heard so much about, especially recently. It's all about making lots of copies of DNA, right?
Precisely. PCR is like a molecular photocopier, or maybe an amplifier is a better word.
Okay.
It allows scientists to take a very small amount of DNA, maybe from a crime scene or a fossil, or just a tiny sample, and create millions, even billions, of identical copies of a specific DNA sequence.
Wow, billions.
Yeah. This massive amplification makes it possible to study DNA in great detail. It's incredibly valuable for detecting genetic markers, diagnosing genetic disorders, and even analyzing DNA from ancient samples where there's very little left.
Then there's DNA fingerprinting, which I always associate with forensic science and crime scene investigations.
You're right, that's a major application. DNA fingerprinting focuses on analyzing highly variable regions of an individual's DNA areas where the sequence differs quite significantly from one person to another. They're called STRs, short tandem repeats, usually.
Highly variable, meaning more unique.
Exactly. By examining these highly polymorphic regions, scientists can create a highly specific genetic profile. And as you mentioned, it's a powerful tool in forensics for identifying individuals, also in paternity testing to establish biological relationships, and even in wildlife conservation to track populations and fight poaching.
So the core idea behind all of this is that we inherit half of our DNA from each parent, creating this unique genetic blend that is specific to us, unless you're an identical twin.
That's the fundamental principle. And by analyzing specific genetic markers within that inherited DNA, we can not only predict certain physical traits, eye color, hair color, maybe predisposition to certain diseases,
Right.
but also achieve very accurate individual identification. This has truly revolutionized fields like forensic investigations, as we said.
It's remarkable how these advancements have had such a broad impact across so many different areas: medicine, law enforcement, basic research.
It really cuts across disciplines.
Now let's move on to a topic that sounds like it's straight out of a science fiction novel: gene therapy and genome editing.
Ah, yes. This is where molecular biology offers the real potential for transformative medical interventions. It's incredibly exciting and also raises some profound questions.
So what is human gene therapy at its core?
At its core, it involves manipulating the genes within a person's living cells for the purpose of treating disease. The goal is to correct the underlying genetic causes of illness.
How do you do that?
Well, through strategies like replacing faulty genes with healthy copies, or inactivating disease-causing genes that aren't functioning correctly, or even introducing entirely new genes that can help fight the disease.
And our sources mention a wide range of diseases being targeted: cancer, genetic disorders, infectious diseases.
Yes, the potential applications are vast. Researchers are investigating gene therapy for a whole spectrum of conditions.
What are the different kinds of tools or products used for this gene manipulation? It sounds complex.
There are several different types or vectors being developed and tested. You have plasmid DNA. These are engineered circular DNA molecules that can carry therapeutic genes into cells.
Okay.
Viral vectors are very commonly used. These are viruses that have been modified, basically stripped of their disease-causing parts, and engineered to safely deliver genetic material into cells.
Using a virus to deliver a cure? Interesting.
It sounds counterintuitive, but they're very efficient delivery vehicles. Then there are bacterial vectors, using modified bacteria similarly. And then you have the human gene editing technologies themselves.
Like CRISPR.
Like CRISPR, yes, which aim to directly modify the patient's own DNA to disrupt harmful genes or repair mutated ones. And finally, there are patient-derived cellular gene therapy products.
What does that mean?
It means taking a patient's own cells, say, immune cells out of their body, genetically modifying them in the lab to make them better at fighting cancer, for instance, and then reintroducing them.
Wow. It sounds incredibly promising, especially for diseases that were previously considered, well, untreatable.
That's the hope, absolutely. For many inherited disorders or difficult to treat cancers, it offers a potential path forward where none existed before.
And then there's CRISPR-Cas9, which has been getting so much attention. You mentioned it. It's described as a real game changer in genome engineering. What makes it so significant?
CRISPR-Cas9 is indeed revolutionary. The reason is its remarkable precision and efficiency in making very specific changes to DNA within living cells and even entire organisms.
Precision and efficiency.
Think of it as a highly targeted molecular scalpel guided by an RNA molecule to a precise location in the genome. It can cut, delete, or even insert genetic material with unprecedented ease and accuracy compared to older methods.
So it made gene editing easier.
Massively easier, faster, and cheaper. This had a profound impact on basic research almost overnight. It allowed scientists to manipulate gene expression, study the epigenome, those chemical modifications to DNA, conduct genome-wide screens to understand gene function, even visualize chromosome organization. CRISPR didn't just provide a new tool, it sort of democratized genome editing.
Putting that power into many more hands.
Exactly. It accelerated discovery at an unprecedented rate.
But it's not just a tool for basic research anymore, right? Our sources indicate it's really starting to move into real-world applications.
Absolutely. CRISPR tech has shown significant promise in correcting genetic disorders in animal models, which is a key step.
And human trials.
And now it's being evaluated in clinical trials for human diseases, particularly for certain inherited eye diseases, blood disorders like sickle cell anemia, and also for developing more targeted cancer therapies.
Where are these trials happening?
There are ongoing clinical trials in various parts of the world, including China and the United States, among others. It's a rapidly moving field.
And its potential reaches far beyond just medicine, doesn't it? I remember reading about applications in speeding up breeding of crops and livestock.
Yes, huge potential in agriculture, making plants more nutritious or drought-resistant, for example.
Developing new antimicrobial agents, which is critical with antibiotic resistance.
A major global challenge, yes. CRISPR could help there, too.
And even controlling populations of disease-carrying insects like mosquitoes through something called gene drives.
Gene drives are a powerful and also controversial application. But yes, the potential uses seem vast.
It seems like widespread adoption of CRISPR across diverse sectors, agriculture, environment, public health, could address a multitude of global challenges.
The potential is immense, truly. But again, it comes with ethical discussions we need to have.
Definitely. Okay, now, another area where molecular biology is making really significant strides is in how we detect those pesky pathogens, viruses, bacteria that can make us sick.
Pathogen detection, yes. Crucial for public health.
It sounds like the traditional methods we've relied on have some inherent limitations.
That's correct. Traditional methods, while valuable, often have drawbacks. They can be time-consuming waiting days for a culture result, for instance.
Right.
They might not be sensitive enough to pick up very low levels of a pathogen early on. They might lack specificity, confusing one bug for another. They can be expensive, require highly trained technicians, and sophisticated lab equipment that isn't always available everywhere.
So things like using restriction enzymes, sequencing, PCR, they all have some downsides.
In certain contexts, yes.
Yeah.
PCR, for example, is powerful, but usually requires specific lab machines and temperature cycling. Isothermal amplification methods try to address that, but everything has trade-offs in speed, cost, sensitivity, or specificity.
But it sounds like CRISPR-Cas technology is emerging as a real game changer in this area as well, not just for editing, but for detecting.
Exactly. This is another exciting application. CRISPR-Cas systems can be programmed to recognize specific DNA or RNA sequences unique to a particular pathogen.
How does that help detection?
When the CRISPR system finds its target sequence, it triggers a secondary reaction, often releasing a fluorescent signal. This offers the potential for significant improvements. Faster results, higher sensitivity, and very high specificity, telling you exactly which pathogen is present.
Our sources provide some specific examples like Sherlock and Detector. What makes these CRISPR-based diagnostic tools so effective?
Right, Sherlock and Detector are two of the pioneering platforms. Sherlock, that acronym stands for Specific High Sensitivity Enzymatic Reporter Unlocking.
Catchy.
Yeah. It's demonstrated the ability to rapidly detect viruses like Zika and dengue, sometimes in just a couple of hours, and with incredible sensitivity down to detecting maybe just one copy of the viral genetic material in a tiny sample.
Wow.
It's also been adapted for West Nile, yellow fever, and importantly, SARS-CoV-2. Detector, that's DNA endonuclease targeted CRISPR transporter, is similar. It's also been successful with SARS-CoV-2 and has even shown the ability to differentiate between different high-risk strains of HPV, like HPV-16 and HPV-18.
Being able to tell different strains apart seems really important.
Crucial for treatment and prognosis in many cases, yeah.
And it sounds like researchers are even developing CRISPR-based chips that could potentially make pathogen detection even faster by eliminating the need for that initial DNA amplification step, the PCR part.
That's the next frontier. These amplification-free methods aim to streamline the diagnostic process even further. The goal is quicker, more user-friendly tests, maybe even suitable for point-of-care settings or resource-limited areas.
So CRISPR is becoming this versatile molecular Swiss Army knife.
You could say that. Beyond diagnostics and the editing we discussed, it's a vital tool in labs for understanding gene function, correcting defects, enhancing traits in various organisms, and people are actively exploring its potential for directly combating viral infections within human cells, too.
So it seems like CRISPR-Cas isn't just about editing genes, it's also becoming indispensable in identifying the very things that can harm our genes and our health.
A powerful diagnostic tool with huge future potential, yes.
Okay, now let's venture into a realm that sounds almost like pure science fiction becoming reality. Molecular robotics.
Molecular robotics. Yes, this is a really fascinating interdisciplinary field. It pulls together ideas from supramolecular chemistry, bio and nanotechnology, and even computer science and informatics.
What's the big challenge there?
One of the significant challenges has been figuring out how to control a large number of these incredibly tiny robots simultaneously. How do you get them to work together in a coordinated way to perform complex tasks and achieve what are called emergent functions?
Emergent functions.
Complex behaviors that arise from the collective interaction of many simpler components, like how ants build a complex nest, even though each individual ant follows simple rules.
Got it. So in this context, what exactly defines a molecular robot? Are they like tiny metal machines?
Not usually metal, no. Think of a molecular robot as an integrated system built from individual molecular components. These components can act as processors or logic gates to make simple decisions.
Like a tiny computer.
A very, very simple one, yes. They can also act as sensors to gather information about their immediate environment, like detecting a specific chemical. And as actuators to carry out specific actions like releasing a drug molecule.
And they operate on their own.
The ultimate goal is for these robots to operate autonomously through molecular computation, responding to their surroundings and performing tasks without direct external control at every single step.
And what kind of molecular building blocks are we talking about? What are these processors, sensors, and actuators actually made of?
Often, DNA and RNA are key building blocks. DNR-RNA nanostructures are being engineered to function as molecular sensors. They can detect specific molecules or changes, like pH, and convert that information into an output signal.
Okay.
Photoresponsive molecules, which change when hit by light, can also be sensors. For information processing, DNA-based devices like seesaw gates show promise for performing basic logic.
Logic gates made of DNA.
Yes. And for actuators, the parts that do the work, researchers often use naturally occurring biomolecular motor proteins.
Like the ones in our muscles.
Exactly, like actin-myosin or microtubule-consanguinine systems. They are incredibly efficient at converting chemical energy, like ATP, into mechanical motion or force.
It sounds like we're getting better at designing these complex molecular parts for sensing and acting.
We are making progress, yes. Designing complex structures with specific functions for sensing, signaling, actuating.
But the real hurdle is still being able to scale up and effectively control large numbers of these tiny machines working together.
Precisely. Scalability and control remain major challenges. How do you coordinate millions or billions of these things? This field also leans heavily on advances in DNA reading, editing, and writing technologies, next-gen sequencing, CRISPR, DNA synthesis, to create these nanoscale structures.
Our sources mention the evolution from relatively static systems, like DNA origami structures,
Where you fold a long DNA strand into a shape, yeah.
to these more dynamic DNA bricks that can incorporate tens of thousands of unique DNA strands. That's just an incredible level of complexity at such a tiny scale.
It truly is. The beauty of using DNA is the inherent reliability of base pairing A with T or U, C with G. It gives you a very predictable and robust way to build intricate nanoscale structures and program dynamic systems.
And things like primer exchange reactions, PER. What's that?
PER is a really clever mechanism. It allows for the controlled assembly of new, longer DNA sequences from smaller, pre-designed fragments, often using hairpin shapes and short primer strands. Crucially, these reactions can be designed to be signal-dependent.
Meaning they only happen under certain conditions.
Exactly. They might only occur in the presence of a specific trigger molecule, like a particular piece of RNA found only in cancer cells, for instance.
So these molecular robots could potentially be programmed to perform very specific tasks, like targeting and destroying specific RNAs, or even recording the order of biochemical events.
Yes, precisely. Recording event order, assembling structures only when needed, and techniques like autocycling proximity reporting, or APR, use PER to map the 3D geometry of nanoscale structures.
How does that work?
You attach DNA hairpins to different target molecules. The hairpins are designed to react and link together only when the targets come very close. Then you sequence the linked DNA, and that tells you which molecules were close to each other, mapping the shape.
Ingenious. And toe-hole switches, what are they?
Toe-hole switches are another clever RNA-based tool. They're hairpin structures designed to control cellular machinery, specifically translation-making proteins.
Ow.
They stay closed off until they bind to a specific target RNA molecule. Binding causes the hairpin to open up on, revealing a hidden gene sequence that the cell's ribosomes can then translate into protein.
You can turn protein production on only when a specific RNA is present.
Exactly. It gives you precise control.
And these toe-hold switches can even be engineered with logic gates, like AND or not, similar to computer programming, for really fine-tuned control inside a cell. That sounds incredibly sophisticated.
It is. This level of engineering allows for very precise control. For example, you could design a system that only makes a therapeutic protein if both marker A and marker B are present in a cell.
Wow.
Or maybe build synthetic circuits that act like an artificial immune system, detecting problems and triggering responses, like cell death for cancerous cells. The potential to revolutionize our control over biological processes is truly immense.
Okay, let's look at some specific applications and challenges our sources highlighted. First, DNA loops as calipers. What are they measuring?
These are literally like tiny molecular measuring tools. DNA loops are attached to microscopic beads, and they act as adjustable rulers or calipers at the molecular scale.
What do they measure?
They can precisely measure the size, overall structure, and even the stiffness of other molecules, like proteins. This basic ability to measure at the nanoscale could lead to new, maybe cheaper, tests for protein recognition and analysis.
Interesting. And what about advancements in DNA origami? Sounds like making these structures getting easier.
Yes, big strides there. Instead of needing hundreds of short DNA strands to assemble a structure, newer methods allow folding complex 3D nanostructures from just one single long strand of DNA, guided by shorter staple strands.
Why is that better?
It simplifies manufacturing. It also opens up possibilities for producing these large nanostructures using materials generated inside living organisms, which could be more biocompatible.
It's really encouraging to hear that some molecular robotics projects are moving from academia to the commercial sector. What real-world applications are emerging?
One good example is DNA paint. That stands for DNA-based point accumulation for imaging in nanoscale topography.
DNA paint, okay.
It's a low-cost alternative to traditional super-resolution microscopy. It uses transient binding of fluorescent DNA strands to achieve incredibly high resolution, down to just a few nanometers. You can precisely see where individual molecules are in a cell.
Exchange paint.
That builds on DNA paint. It allows for highly multiplexed imaging looking at many different target molecules simultaneously in the same cell, or even in thick tissue samples. You get a much richer picture of what's going on.
The incredibly small size of these molecular machines seems like a huge advantage. You could have billions of them working on a task.
Absolutely. That inherent scalability and the potential for mass production is a key benefit. Imagine billions of tiny devices targeting cancer cells or repairing tissue.
But coordinating them all, that's the tricky part.
That remains a significant hurdle, yes. Orchestrating the interactions of billions of individual molecular machines to achieve complex collective behaviors, like relaying information reliably or cooperating on a construction task, is still a major research challenge.
And then there's this interesting macroscopic approach with kilobots. These aren't molecular scale, right?
No, kilobots are much bigger inch-long robots, but the idea is related. Researchers use large numbers of them, sometimes thousands, to study principles of collective behavior and self-organization.
Learning lessons for the molecular scale.
Exactly. How do simple rules lead to complex group behavior? Understanding this with kilobots might inform how we design and control swarms of molecular robots eventually. It also opens up new avenues in larger-scale robotics and automation.
So as we wrap up this incredible journey through molecular biology, from Mendel's peas to molecular robots, what are the ongoing evolutions and key challenges the field is still grappling with?
Well, despite all the amazing progress, there's a continuous global demand for refining methods. We always need better tools.
Like in cancer research.
Exactly. Cancer research is a prime example. We need more precise methods to identify subtypes accurately, predict treatment response for individual patients, true precision medicine.
And studying really complex systems.
That's another huge challenge. Systems like the human brain, with its trillions of connections and intricate interactions, understanding them at a molecular level is incredibly difficult.
And all the data being generated, it must be overwhelming.
Absolutely. The explosion of genomic, proteomic, transcriptomic data, all the omics is fantastic. But we need increasingly sophisticated computational methods for data analysis and integration to make sense of it all, to find the meaningful patterns.
And the ethics, especially with gene editing.
That's paramount. As the power of technologies like CRISPR grows, rigorous ethical considerations and public dialogue are absolutely essential to ensure responsible development and use.
And just because something works in the lab doesn't mean it helps patients right away, does it?
That's a critical point, the translation gap. Bridging the gap between lab discoveries and effective, accessible clinical applications for patients is a huge ongoing challenge. It requires collaboration between scientists, clinicians, regulators, and industry.
Which points to the need for working together globally.
Definitely. Fostering robust global collaboration and open sharing of information and resources is essential. These challenges are too big for any one group or country to solve alone.
So, to bring it all together, it's clear molecular biology is this incredibly dynamic, rapidly advancing field. It's reshaping our understanding of life itself.
Fundamentally.
And giving us powerful new tools to diagnose, treat, and maybe even prevent diseases, plus building these amazing nanoscale machines. We've covered so much ground in this deep dive, from basic inheritance to gene editing and molecular robots.
It's been quite a journey.
Considering the remarkable speed of these advancements, what do you think will be the most significant societal impact of molecular biology in the next decade? Really gives you a lot to think about, doesn't it?
It truly does. The ongoing exploration holds immense potential for even more profound insights and transformative applications. The journey of discovery is far from over, and the possibilities ahead are, well, genuinely exciting and maybe a little daunting.
Thank you, viewers, for watching. If you found this video helpful, don't forget to hit that like button and share it with your colleagues. And if you haven't already, make sure to subscribe to the channel by clicking that subscribe button below, so you never miss out on valuable insights. I'm Brian Edmonds, signing off. See you in the next one.